So if you could just introduce yourselves a little bit for our audience and what it is you research.
Well, my name is Fernando Guzman. I'm from Mexico, and I did my PhD in synthetic biology in the University of Groningen in the Netherlands. And after I moved to perform a postdoc in James Hasselhoff Lab in the University of Cambridge. So in this place, I started to work in a consortium to try to develop a low-cost diagnostics device for low and middle income countries. But after the pandemic, we have to change a bit. So basically, I'm doing synthetic biology.
Cool. We invited you today specifically after reading your paper on the Cellfree systems and reducing the cost of the Cellfree systems for low-income countries. Could you introduce the idea of the Cellfree system and what those are exactly?
Well, the Cellfree system is basically an in vitro system to produce proteins. Some they call TXTL, which means that is the transcription and translation of the proteins. So basically, it's a system that doesn't involve cells, just extracts or purified proteins to produce a target protein. And this target protein you can use for different applications.
Okay,
what would be the benefits of producing proteins using this system?
Okay, to produce proteins with Cellfree, it allows to easy characterization because it's easier and faster if you compare with the traditional approaches that to make a recombinant proteins. Normally it takes like 2 weeks. In using Cellfree, it just takes a couple of days. And the fact that you use a really small amounts, you reduce cost. But most of the times it's really expensive because you have to rely in system that you have to buy through the companies. So it's always nicer if you can produce your own extracts or your own Cellfree to work with.
What are the sort of hurdles that you have to overcome to produce your own extracts? Like why is it so expensive? That's it.
Well, there is many versions to produce Cellfree. So the main difference of them is the energy solution, which contains phosphate toners, and most of the times are really expensive. So one of the most uses is PEP, phosphoenol pyruvate, creatine phosphate. And if you compare when you buy if you want to buy 1 gram, for instance, it costs around 200 pounds, just 1 gram. It's really expensive to. So the first, if you want to develop a low cost, I think that you have to invest a bit at the beginning. The other part that is also expensive is the nucleotides. So the nucleotides represents the half of the cost. Other thing that is expensive is for instance, in a low and middle countries, specifically in countries that are in Africa, they don't have ultra freezers, so they cannot produce cell extracts and store them. So that is a really a huge disadvantage. So that is the main cost.
So just to clarify, you would say there are 3 main sort of components that are quite expensive in this, which are the sort of the extracts that you use, the nucleotides, and finally the ultra freezers, which are unavailable in certain specific low-income countries.
Yes, but there is in the mix. There are other cofactors that are also expensive, but we know from some time ago that they are not really necessary. For instance, the tRNAs they are really expensive, but most of the time you don't have to include it because they have studies. There are studies that shows that they are not really necessary, but they are expensive because you can buy just 250 milligrams and from the companies, and yeah, it's expensive.
And I'm just wondering, in your recently published paper about reducing the cost of Cellfree systems, how did you go around reducing the cost? Like, what were the novel approaches that were developed for the paper?
Well, in the paper, basically, we were target low and middle income countries, but it can apply from every lab that I wanted to use. Our first idea was that, for instance, when you do the research in these countries, most of the times you don't have access to proteins. The main goal is why is this happening is because, for instance, in the UK, you have the companies that are in the country. So you buy directly the enzymes or other proteins that you needed. In these countries, you don't have them. So you have intermediaries, and this increment the cost. And imagine that, okay, you can get a budget, you have enough money to buy them, but most of the times are for 3 times or 4 times more expensive. So it's increased and the budgets are less. But it's not only this is the problem. The problem is also that, okay, you have the money, but you buy the regions, but it takes like at least 1 month to arrive to the country because they have to order to these countries that they where the companies are placed. So that is the main problem. So we imagine we were thinking that if we produce a cell-free extract that you can store dry, you can dry and store, and after, if you need, for instance, an enzyme, you can produce it really easily. Just mix the a DNA that encodes for this enzyme plus the cell extract that you dried, and you just incubate for overnight, and the next day you have your fresh enzyme. So imagine this reduce cost, time, and this speed up the research. And we are really lucky because, for instance, Jenny Malloy and Jim Cancelov has this initiative of free genes. Some of them has been deposited in collaboration with the University of Oxford. So basically, you can order these synthetic genes that are already cloned in a vector, and you do some cloning, put some promoters, and you just mix with the cell-free and produce your protein. The advantage of this initiative is that it's totally free, so they send you with any charge in any part of the world that you need. So taking this, initial ideas, okay, we have to produce a system that is really cheaper if we compare with the others. For instance, in the paper, we found that if we choose a different company, so for instance, to buy this PEP, the phosphoenolpyruvate, most of the times our first option is the huge companies, but there are really small companies that they sell really high quality compounds. So we found that, for instance, Alpha SR, that is a company in the UK, it sells 5 grams for really nothing, so like 60 or 80 pounds. So, but 5 grams is a lot. So this was the first improvement that we did.
What was it? So you mentioned that this can be used, I think you mentioned it was for diagnostics, your research was particularly interested in. How has this been applied for a diagnostic purpose in these low or middle income countries? The cell-free systems, that is. Well,
after the pandemic, many people start looking around cheap devices to do diagnosis. And one of the main tools, and many people knows, is the LAMP reactions. LAMP is isothermal reactions that involves 4 or 6 primers, and it's amplified the target sequence. So how we can apply this? If you buy this LAMP, this polymerase in the companies, you have to always to depend to these companies to do your diagnostic tool. But if you use cell-free, you can easily produce this protein or this polymerase that you are going to use for diagnostics. So you are really in a way you are independently. And the good thing is for the lamp, this polymerase is off patent, so you you don't have to deal with these things, so you can use whatever you want. And this is one approach. Another approach is that you can also produce Cas proteins to produce this protein and do some system like detector or Sherlock to detect the to do diagnosis for a virus or a bacteria. Actually, there is a paper that before this that we show that we can produce a really low cost extracts to detect a potato virus, and this is a proof of concept that you can use to characterize these kind of tools, because this we took the initial idea was took from a paper that they use a pure system. Pure system is another cell-free system, but it involves that you have to purify each of the 36 proteins that you need to the transcription and the translation, which is really annoying. And if you buy in the company, it's really expensive. For instance, one reaction costs like 20 pounds. So in this paper, we show that not really LA system, so crude extracts that basically cell-free, and you can use to characterize these tools for diagnostics.
Okay.
And
Go ahead.
well, I was just wondering, how does the methods that your lab developed compare to the traditional cell-free methods, or the traditional or commercial mixes?
Well, this is was very interesting, because when we try to reduce the cost, we didn't know that we are gonna find something that is really useful and nice to improve the cell extracts in a cheaper way. So for instance, the first is, instead of use a professional freeze dryer, we see that we can use a desiccator just with silica, and basically just mix with some sugars, you can get the same yields of proteins if we compare with the cell-free reactions that we're drying using this professional freeze dryer. Other thing that we found is that if we supplement the cell extract with lactose, you can boost the reactions using maltodextrin, which is the cheapest energy source that you can use for cell-free. But the disadvantage of using maltodextrin is that the yields are just the half of PET. So that is why some people they don't like to choose maltodextrin against or if they can use PET. But if you add lactose, you can get these yields. And another approach that was really new that I think that so far I don't know on that other groups reports, is that the endogenous regeneration or biosynthesis of nucleotides, because most of the times these ones are really is one of the main elements of the cell-free reaction, the nucleotides. And there were other studies that show that they can use, instead of triphosphates, a monophosphate nucleotides, and it reduced the cost. But you still using nucleotides. So when we were removing each of the elements, we discovered that at the end, that even when we remove all of them, we saw some activity or some protein that was producing in the PET system. So taking in this this view or this curious thing, we decide to add the lactose that we knew that it can improve, and it improves, and they give you around the 40% of the reaction life, which is really good, because if you want to use the Cellfree for educational purposes, you don't need high amounts of proteins, because if, for instance, if you use fluorescent proteins, just you want to see the colors, and it don't make sense to use a really full mix to do it. So we show that you can use the ultra low cost Cellfree system, as we call it, and to produce these fluorescent proteins for education.
Hmm.
And how would you say these ultra low cost mixes compare with the mixes sort of available commercially currently in terms of performance, for example?
In terms of performance, is the 40%, because our cell extracts are basically in the same level that the commercial extracts. For instance, we compare the Mighty XTL, that is like one of the most used commercial Cellfree available, and it is based on maltodextrin. And actually, when we add lactose, we also improve the commercial version. But okay, the thing is that if we compare with the commercial version, the ultra low cost is 40%. If we say that the commercial is the 100% in efficiency and yields.
So like your sort of method would be sort of 40%, whereas and the commercial would be like 100% in that kind of comparison.
Yes.
Okay.
Yes. Yeah, but for instance, for fluorescent proteins is really good, because in the paper we show that you can see the fluorescent even by eye, and after blue light, you can also see them. And as I say, for education, I think that is enough.
Yeah. Do you see other applications of this? Like what beyond sort of education and diagnostics? Is there others that you can think of?
Well, no, our next challenge is to use Cellfree to, as I say, to produce molecular biology reagents, more proteins. We didn't put them, but we planning to produce, for instance, this 7 RNA polymerase, which is one of the most expensive reagents, and is particularly used, for instance, for when you want to test in vitro the Cas proteins. You have to produce RNA, so that is one of our goals, to try to produce this. And also another proteins for another isothermal amplification method, that is RPA, that involves 4 or 5 proteins. So basically, yeah, that is, and you can use these proteins for whatever purpose that you wanted to use.
And what do you see as sort of, what do you view as the sort of most significant challenges in making this sort of this type of mix sort of more widely used, or used in the places you want to use it in?
I think that okay, you wanted to use, or you wanted to make in the place that you are going to use, I think that you need some experience, because all of them needs, for instance, amino acid solutions, and you have to add one by one if you want to have a really low cost. And also you have, so basically you need experimental skills to produce the extracts. But if you want to send them to another lab, dried samples, for instance, I think that the main problem could be in each country the customs, because some of them are really easy to handle this stuff, but they requires a lot of paperwork. Yeah, I think that this is main change. So, for instance, if a new researcher or a school wanted to use, they have to. The first has investment, and they need, for instance, people with experimental skills to produce them. But actually, if somebody show you how to perform, to make all the solution, it's really easy, because, for instance, the amino acid solutions, you can just prepare once, and you have a lot for years, for years. And but the cell extracts are the more challenging if you want to keep the reproducibility. So that, I think that is the main.
If you could sort of have the ideal scenario, what would you, so let's say 10, 20 years down the line, your research has gone the way you want it to go, what would cell-free systems look like for you?
Actually, it's very, I think that it's a really good question, because the Cellfree is really an old method, so was developed in the 60s, and actually using Cellfree, the people discover the colon use from the, so, and but the people stop to use it because they don't have at this time all the tools that we have now. With these tools, I mean how to cloning, how to design the constructs, and the synthesis of synthetic DNA. So I think that in the next 10 years, the Cellfree is going to be more common to use, to approved. And I think that now we are in a time that the synthesis of synthetic DNA is becoming cheaper and cheaper. So I think that at this time you are going to be able to produce or test more crazy ideas.
Do you think that cell-free systems might replace the classical protein expression, what do you call it, like where you normally grow your cell in an incubator and that sort of thing?
Not really. Well, in my opinion, no, because the yields. So basically, this is like a screening method. So you are gonna spend a lot of time to produce the proteins, but after you find that this protein is working, for instance, for diagnosis or for the purpose that you want to research, maybe you can go back to for the strain, grow the cells and purify the proteins. But for some examples, you are going to depend always on cell-free, because there are proteins that are really toxic to produce in a live system. So they are not going to replace, but they are going to work in parallel.
Okay, I see. Yeah. Right. I think that's all the questions I had. What about you, Sarah and Sandra?
Yeah, I think those were all the questions we had as well.